Okay. So, all right, couple quick reminders. Yes, because you guys are asking about the timing and some stuff. What we have is tomorrow, you have a content worksheet due and a pre-lab worksheet due. Anything else? No, that's it. Okay. Jada looks scared for a second. All right, so you have a pre-lab on Brightspace, you have a content worksheet on Brightspace that is due tomorrow. The other thing that you have, though, coming up is what is a week and a day from, what's basically a week from today? The Mythbuster presentations are this coming Tuesday's lab, not tomorrow, but next week. That is going to be Tuesday, October 7th. Okay. Remember, those are 15-minute oral presentations. Please put them together in PowerPoint. You are going to project that here at the front of the room, and you're going to give that presentation to the class as a whole. I do expect you guys to have some professional literature citations of that. What did I say? I think I said 5. 3 to 4? 3 to 5? Okay, I definitely said 3. You're right. I did. I remember that now. So at least 3 professional sources of information, and those would be journal articles. So, okay. So those are also a requirement for this particular assignment then as well. I want you guys to be going through some quality kind of peer reviewed types of studies to help illustrate your points on your presentations.
Will I expect like the presentation for some of the info, like us to have memorized it and not it be like all just projected on the PowerPoint?
If you are, I find that if you're just reading straight from the PowerPoints, it is not, it kind of ruins the professionalism of the presentation. So when I find somebody's reading from the PowerPoints, they usually are doing one of 2 things. They're like sitting here staring at the computer, or they're sitting here staring at the presentation with their back to the audience. Back to the audience is bad. Just to let you know that one straight off, yes, you would miss a little bit of points if you've got your back to the audience on a regular basis. You're presenting to the class. You should be maintaining eye contact as a good oral presentation technique. The other thing is, I find that if you're just reading from the slides, it gets robotic. It gets a little low energy, and at the same time, it goes very, very quick, somehow simultaneously, which all of that is, again, you're not connecting well with the audience. And then the audience has more trouble following along with the presentation when you're going too quick as well. Okay, questions about that? So there is some oral presentation and some good kind of oral presentation delivery and communication skills that is built into the grade as well. Right.
So this is gonna be like sitting here paying attention, or do we have to fill out?
You not have to fill out anything, but I do expect you to ask questions of other presenters. 2. And they can be during the presentation if you guys are comfortable with that, or just save it for after the presentation. I'll leave that up to the presenter's discretion. Some people don't like to be interrupted.
You have a question for each person.
For all the presentations.
They all.
No, 2 questions total over the course of all the presentations.
2 questions
No.
during the day.
Okay,
During the day.
Got it. Okay.
Thank you, that's better.
At some point between 8 AM.
Yes. All right.
Questions.
This also is going to lead into, remember, there's going to be then a lab quiz on the Tuesday after fall break. And then the second exam is the Tuesday after that. So we got the presentations on the 7th, we have a lab quiz on the 14th, and we have the exam on the 21st. Does that make sense?
Yeah.
Okay, so I tried to keep things spaced out nicely.
Yeah, I'm so glad that you're not having a test. Before fall break.
That's
Everyone else is.
I know that, and that's part of the reason I moved it to after. Technically speaking, it's the movement of having it in the lab periods, which I know is a little unusual, but I'm not the only one that does that. Am I?
Dr. H.
Yeah. But part of having it on those days allowed me to also push it back to fall break a little more comfortably. All right, today we're going to keep on talking a little bit about control of microbial growth. So last lecture, I started to introduce this idea where you have sometimes what are called growth optimums. This is the version of an environmental parameter at which a bacteria grows the best, where it has a maximized growth rate. Environmental parameters could be something like, for instance, temperature or pH. But at some point, there's going to be an optimal temperature where they grow the fastest, or an optimal pH where they grow the fastest. But we can also exploit things like growth maximums and growth minimums. Growth maximum is the maximum value for a parameter at which they will still grow. If you go higher than that for that parameter, the bacteria doesn't grow, or even sometimes it dies, for instance. If you go below a minimum, again, it will not grow. Its growth rate reaches 0. So if you go lower, it's not going to grow, or it may even die, for instance. And we started talking about different examples of parameters such as temperature, osmotic conditions, oxygen concentration, radiation, pH. And we're going to talk about each one of these one at a time. Then we're going to start off with temperature today. Then temperature, we can use temperature to limit bacterial growth. And we do this pretty often. For instance, if we raise the temperature, especially up to things like cooking things or boiling things, we can then kill contaminated microorganisms. And we do that to get rid of any microorganisms we might think are worried about, for instance, causing disease. We can do things like pasteurization. Do you guys know what pasteurization actually is? So what do we pasteurize? What kind of milk? Pasteurization is holding milk at high temp for a long period of time. But you can't boil milk. You guys know why you can't boil milk? Huh? It curdles it. So if you do milk to too high a temperature boiling it, it actually causes a bunch of the proteins in it to denature, then they tangle up and they curdle. Okay, so you can't boil milk to sterilize it or anything like that, because then the milk's going to be gross. Once again, what ends up happening is you hold it at high temp, but you hold it at high temp for a long period of time. That abnormal conditions is enough to kill microbes in there, especially the kinds of microbes that cause disease and food poisoning from like contaminated milk. Okay, so that's what the pasteurization is. You can do things like lower temperature. For instance, we use refrigeration to restrict microorganisms. So again, we can go beyond the growth maximums or go below the growth minimums when it comes to temperature. Heat, if you go up too high, why does heat affect cells? Well, especially if you do things like cooking or boiling, high heat. For instance, what it can do is it can take proteins that are under their normal functional shape, and then when you heat a protein, it unfolds, or what's the name for that? Denatures the proteins. So again, a denatured protein is a unfolded protein, is really what we're talking about there. And if they don't have the right shape, are they functional? No. So they might fold up to the correct shape. Now you got a functional enzyme, but you heat it, it denatures it, it unfolds, and it's no longer functional anymore. So it can cause things like high temp, will cause things like the enzymes in the cell to not work. But even worse than that, sometimes you go up to boiling temp, and it can cause double-stranded DNA to become single-stranded DNA. What kind of chemical bond holds together the two strands of DNA? Hydrogen bonds. AT, hydrogen bonding, GC, hydrogen bonding. Hydrogen bonds, weak or strong? Weak, and pretty weak. And for instance, just heating something up to boiling is enough to disrupt those hydrogen bonds. And now again, the DNA comes apart from double-stranded DNA held together by those hydrogen bonds, breaks those hydrogen bonds at a high temp, and you got single-stranded DNA. That's really bad for cells. Okay, the DNA's damaged, that's going to be bad. It can also cause membranes to melt. I don't have that up here, but membranes can get disrupted and melted at high temp. So heat can, again, denature proteins. It can melt apart DNA to single-stranded DNA. It can disrupt membranes, and those are all bad for cells. Cold can cause membranes to freeze and then shatter. So at cold temperatures, the membrane loses fluidity and it will freeze in place. And then any kind of pressure on that membrane can cause it to crack open. Think of it as stepping on thin ice, then, for instance. So cold can cause membranes to crack and break open. They can make proteins inflexible and cause them to denature. So sometimes cold temps can also cause protein to denature. A bigger problem is usually a protein comes along, and we're gonna talk about this later. But if you have a substrate, a reactant, usually the enzyme comes along, binds to it, and then upon binding, the enzyme will change shape. And then it puts stress on the reaction, the reactant kind of almost squeezes it. But in a cold, the protein kind of hits it, has trouble changing the shape, and it can't speed up the reaction properly. So when I say proteins being inflexible, they can't change shape, which is often necessary for them to do their jobs. And it just causes chemical reactions to be slow. So metabolism is just all the chemical reactions inside the cell. But again, colder temps, what happens to reaction rates? They also slow down. And that's going to limit the chemistry in the cell. We can use things like very high heat to kill things. Stuff like boiling is not sufficient to kill all bacteria, especially if bacteria make endospores. So we talked about endospores, but as a dormant, very resistant version of a bacterial cell. And for instance, boiling by itself doesn't kill endospores. You can boil samples and the endospores will make it through. You put them back to normal temps, endospores wake up, and this bacteria start growing up again. Okay, this is why we use things like autoclaves. Autoclaves put not just boiling temps, but high pressure. So they do like 15 pounds of pressure above boiling temps, 121 Celsius, for at least 20 minutes. So it's a big, giant, glorified pressure cooker. But the combination of both high pressure and also high temp is then going to be enough to kill even endospores, for instance, and fully sterilize stuff. Now, when it comes to bacterial species, turns out though, different kinds of bacteria have different temperature preferences. So for instance, when it comes to temperature, most bacterial species grow optimally at temperatures between 10 to 50 degrees Celsius. Any bacteria that grows best at these temperatures is called a mesophile. So a mesophile, again, what that is it grows, it's a species that grows best between 10 to 50 degrees Celsius. And that is in Celsius, just to warn you guys, not Fahrenheit. It turns out also most bacterial species are mesophiles. So again, mesophiles are any bacteria that grow best or optimally at a condition between 10 to 50 degrees Celsius. Let's just say 37 degrees Celsius. Most bacterial species on Earth are also mesophiles. Why is this the most common temperature range? Or why are most bacteria mesophiles? Why do most of them grow optimally at this temperature range? Yeah, it's just the most common temp range on Earth. It's just the nice normal-ish temperature. So most bacteria fall in the mesophil category because mesophil temperatures are the most common temperatures. With that being said, there are some bacteria that grow optimally at different temperature ranges. So for instance, thermophiles. Thermophiles grow best at these temps. So they grow optimally where they have their fastest growth rate at temperatures between 50 to 80 degrees Celsius then. So thermophiles, these are bacteria that have a high optimum growth temperature. And so for instance, the bacteria that I talked about in this rainbow pool from Yellowstone National Park, that's a hot spring, sulfurous hot spring, but the bacteria that are living inside that hot spring, very high temp, and there are going to be thermophilic bacteria growing inside of there. And an example of that might be, and for instance, is Thermophilus aquaticus, is a hot spring bacteria that you usually find it living in those environments. They have to have adaptations to live at those high conditions. So they have to have adaptations that help them survive at those high conditions, but not just survive, but thrive and grow best under those high temp conditions. So for instance, they're gonna have thermostable proteins. A thermophile's proteins are gonna fold properly at that high temp range. And therefore, they're gonna work best at that high temp range. They're also gonna have membranes with long fatty acids, straight unbranched fatty acids, and saturated chain fatty acids. Again, we talked about this earlier, but this lowers their membrane fluidity. Their phospholipids don't move around as easily, and therefore their membranes are less likely to melt apart at these high temps. Questions about that? There are some other categories. So set up for this, mostly ocean floor, away from coastline, and you go out there and you're not going to see anything. So you go out to the open ocean floor, and this is well away from the coastlines, deep, deep, dark ocean. It's cold, there's no light, and there's nothing there. And so most of the ocean floor is pretty barren. However, every once in a while, you get these deep sea hydrothermal vents, or what are called black smokers. These are kind of like miniature undersea volcanoes that are constantly erupting. These hydrothermal vents. You got this lot of heat being spewed out from these, some minerals being spewed out from these. And around these hydrothermal vents, you have clusters of life. And suddenly, it just explodes. And for instance, these guys right here are tube worms with some bacterial partners. So the white stuff is animal tube worms. The red stuff is actually bacteria they live in a symbiotic relationship with. Okay. The bacteria eat the mineral nutrients coming out of the hydrothermal vent, and then they share some of the nutrients with tube worms. Tube worms help to protect them and give them a nice place to live. Okay. Now, what you have near these hydrothermal vents is, again, most of the ocean, deep, dark, cold. 4 degrees Celsius, just above freezing. But near these hydrothermal vents, they're pumping out heat, and the water is very hot next to them, up to 300 degrees Celsius sometimes. But then you get a temperature gradient where it gradually falls to the floor. And that temperature gradient is fairly steep. But what you typically get is something like the tube worms or the bacteria. They're not living here. That's too hot. But they're living somewhere in this temperature gradient. And so somewhere in that gradient, you find them. And in fact, you have sometimes what are called hyperthermophiles. These are bacteria that prefer, or prokaryotes that typically live at conditions anywhere from 80 to 113 degrees Celsius, that they grow best optimally between these temperatures. So for instance, there is a type of Archaea species called Pyridictium abyssei, and it grows best at 113 degrees Celsius or above boiling temps. Boiling is 100. Okay, this would be an example then of a hyperthermophile. Questions about that? All right, with these things to keep in mind, something like a mesophile's protein is going to fold up and be functional at things like 37 degrees Celsius. It's going to fold up and functional between that temperature range of 10 to 50. It depends a little bit on what its species is and where it grows optimally. But if you raise it up to say 75, that protein is going to denature, unfold, and be non-functional. Remember, with thermophiles, their protein is going to fold up and be functional at that high temp condition. But what happens to their protein at the lower temps? It denatures and unfolds, and it's no longer functional. Something like a thermophile's enzymes will work properly at the high temp, but if you take them down a low temp, they denature and their enzymes aren't going to work properly. They are adapted to those high temp conditions. And very small changes in the primary amino acid sequence of a protein can make it either thermostable or not thermostable, for instance. Then, okay, questions about that? Question for you then. Can thermophiles cause disease in humans? Show of hands. We're going to say, how many of you think that thermophiles can cause disease in humans? How many of you think they can't? They can't. Because their enzymes aren't functional at normal body temp. 37 is normal body temp, and it turns out their enzymes will denature, unfold, and they can't grow at that temperature at all. If they can't grow at that temperature, they can't cause disease in us.
If they rapidly, like, got into like a human body, could they cause like a very short disease?
No, typically not. You would have to get exposed to like insane amount of them to cause damage in a short amount of time. You got other problems probably in that case. The only way you're probably going to get exposed to a large amount of thermophiles simultaneously is, say, for instance, you fall into a hot spring and then you got a whole lot of other temperatures like you're boiling alive issues instead. So no, probably not. You also have to worry about thermophiles, hyperthermophiles. They also have to worry about their DNA melting from double-stranded DNA to single-stranded DNA. So for instance, normally with DNA, if you raise it up to 90 to 100 degrees Celsius, it melts apart into single-stranded DNA. How might hyperthermophiles stabilize their chromosomes and prevent that from happening? What's some way that you would make the double-stranded DNA held together more strongly? Huh? What? What about the nitrogen spaces? More hydrogen bonds between them would make them hold on together more strongly. How can you get more hydrogen bonds between them? A pairs with how many hydrogen bonds? 2 H bonds. G pairs with C, 3 hydrogen bonds. So typically, when you have something like a hyperthermophile, their GC content of DNA is 80 to 85% or higher. Something like a hyperthermophile uses the bare minimum of AT pairs in its DNA just to code for the necessary genes, but it has a very, very high GC content. So most of the nitrogenous bases in its DNA is G and C. Very few A's and T's, just enough to properly encode their genes. Questions about that? This means more hydrogen bonds, which means it's harder to break the double-stranded DNA into single-stranded DNA then. So they just have what's called a high GC content in their DNA. For instance, mesophiles are usually around 45 to 55% GC. Questions about that? Last temperature category then is psychrophiles. Psychrophiles have metabolism that, psychrophiles live best at temperatures between -5 to, actually, I should put 10. That was a typo. That should be 10. Okay, there we go. Sorry about that. Psychrophiles grow best at temperatures between -5 Celsius to 10 degrees Celsius. They have a metabolism that is optimized for low temperature conditions. Their proteins and their enzymes work best at those low temperatures. They're typically also going to have membranes that have a lot of fatty acids that are unsaturated, short, or branched. And the idea here is those type of fatty acids increase the fluidity of the membranes and make it less likely that the membranes are going to freeze in place. Have you guys ever heard that like fish are rich in unsaturated fats? Well, they are. Where do the fish live? In the water, and the water is usually cold, especially in the ocean. So especially ocean fish tend to have a lot of unsaturated fats in their membranes. Okay. Questions about that? But so do bacteria living at those low temp conditions as well. Example of a psychrophile could be things like snow algae, Chlamydomonas nivalis. This is a glacier, snowbank. But it's pink because of the snow algae that is living in it. Chlamydomonas nivalis, they have this red pigmentation because they are photosynthetic, and that helps them do their photosynthesis. By the way, a lot of glaciers at mountaintops tend to be getting more and more pink in the last couple decades. Yeah, it's climate change and global warming. Because the mountaintops are warmer than they used to be, there's more of the snow algae growing in the snowbanks at the tops of mountains.
Yeah, but why doesn't that happen in like the Midwest? Is the snow just not there long enough or?
Yes. So these things grow really slow. So it's things like glaciers that are around and don't melt off. The algae does have enough time to get up to a density to change the color a little bit to this pink. Things like snows that regularly melt off, that's not going to be the case. They just have really slow growth rates because they also grow at really cold temperatures. That's a good question. Okay, questions then about things like the different temperature ranges.
I guess it's like kind of like a random question in general, but like how do you culture them like thermophiles and like things like that? Can you do that?
They're difficult to culture, but you just have to grow the cultures at their appropriate temperature range.
How would you even like transport it? Like,
Yeah,
what
how
would
would
you
you work
do,
with
like,
that?
like for instance, like the glacier, like what would you do? Like just like
Oh, not a lot of research is done on those because they grow so slowly that they're difficult to do studies on.
Like how slow? Like it takes years or?
Weeks to months to double. So to go through a round of cell division might take weeks to months in some of those cases.
So it would actually like take years for it to.
Yep.
Have enough to be.
Yep. And so they're probably under-researched. You also typically, strangely enough, also have slower growth rates a lot of times in those thermophiles or hypothermophiles, because again, those high temp conditions are not super hospitable to life. So they will grow a little slow for those guys as well. But no, there's been some studies on thermophiles. You can do them from the environmental source, but you can keep them at temp and bring them in and culture them at the high temp. For instance, TAC DNA polymerase. You guys heard of that before? Thermus aquilus DNA polymerase. It's DNA polymerase from hot spring bacteria. And it's temperature stable, which is why we use it for things like PCR, polymerase chain reaction, which is done at high temps. All right. Next thing we're talking about is osmotic stress. Osmotic stress. What is osmosis? What's the definition of osmosis? It is the diffusion of water, but let's be more specific than that. It's the movement of water from areas of, we describe water's movement. Oops, it helps if I spell areas right. Man, I'm like, okay. From areas of, we describe the movement of water relative to the solutes dissolved in it. So it's movement of water from areas of lower solute concentration to higher solute concentration. And my brackets are shorthand for concentration.
It's because the water is actually.
Because it's the water that's moving, not the solutes. But water is going to move from where there's less solutes dissolved into it to areas where there is more solutes dissolved into it. We also have some tonicity terms. Tonicity. Yeah, there's some different terms for this, but it's always relative. One solution relative to a second solution. Okay. So when I say this term hypotonic, the hypotonic solution is going to be any solution that has a lower solute concentration compared to some second other solution. So the hypotonic solution is going to be the one that has lower solute concentration compared against something else. Here in this particular example, I have a environmental solution and I have a cytoplasmic solution. So I have two different solutions here. I've got the solution that's inside the cell versus the solution that's outside the cell in the environment, and I'm comparing those. Which one is the hypotonic solution if my little red dots here are solutes dissolved into?
Not water.
Just talking about which solution, if the solutes are the red dots, which solution, the environmental solution, is that the hypotonic one, or is the cytoplasmic solution the hypotonic one? And so we're gonna do show of hands again, so you gotta pick one. So is the environmental solution the hypotonic solution? Show of hands. Is the cytoplasmic solution the hypotonic one? Show of hands. Okay, it is actually going to be the environmental solution is then going to be, in this particular case, the hypotonic solution, because there's less solutes dissolved into it. And the little red dots, the solutes, are definitely at higher concentration in the cytoplasm. There's a lot more of them. Make sense? Okay. So the hypotonic solution is going to be in this scenario, the environmental solution is the hypotonic solution. It has a lower solute concentration compared against the cytoplasm. What's going to be my direction of water movement? Yeah, the water is going to move into the cell. Remember, it always moves from lower solute concentration to areas of higher solute concentration. Questions about that? But as water moves in the cell, and then more water moves into the cell, and then more water moves into the cell, and then more water moves into the cell, more and more and more, what could happen to the cell? Yeah, it burst open. If too much water moves into the cell can burst open. This is called lysis. And this is also a version of what is called osmotic damage to the cell. The cell can only accommodate so much volume. Too much water moves in the cell and it bursts like a balloon that you overinflated, for instance. Okay. And that's when a cell, anytime a cell bursts open, it's referred to as lysis of the cell. So membranes have to be protected from that water pressure.
What is the limit strength?
We're going to get to that in here in a second. How is the cell then protected from this type of osmotic damage? Actually, a pump is a good idea. So you could have cell pumps. So for instance, something like a paramecium cell might have something called a contractile vacuole, and it pumps the excess water out. But the contractile vacuole is a organelle. Do prokaryotes have organelles? No, so they can't have a contractile. So eukaryotes might have something like a contractile vacuole. Prokaryotes can't do that because they don't have organelles in general. So what's something we have talked about that will protect them from osmotic damage? Huh? It's, yeah. In this case, made out of peptidoglycan. Cell wall is a very, very strong, very rigid structure. It limits the volume of the cell. It can still, so much water can move in, it can break the cell sometimes, but it's hard to do. Okay, but that peptidoglycan structure is protective of osmotic damage. Actually, is also something called Turgor pressure. Turns out almost all cells are pressurized. This is referred to as sometimes Turgor pressure. And most cells have about the same level of pressure as a bicycle tire. But for water to move in, they have to move in with enough force to overcome that pressurized environment. And that prevents water from moving in many cases, or at least too much water from moving in. Again, if they're in a very strongly hypotonic solution for the environment, though, that's gonna have enough force to drive the water in past that turbo pressure point. Questions about that? Hypertonic solution is any solution that has a higher solute concentration compared against some other second different solution. So again, I have an environmental solution, I have my cytoplasmic solution. Which one in this example is the hypertonic solution? I flipped it around. The environment is also the hypertonic solution in this example. I don't have very many solutes in the cytoplasm. I've got a lot more out there around the cell. So in this case, there is a higher solute concentration in the environmental solution, and that makes it the hypertonic solution. So I flipped it around compared to the previous slides. What's going to be my direction of water movement? In this case, it's going to be moving from the cell out to the environment. So the water is going to be moving out of the cell. If a cell is in an environmentally hypertonic solution, then the water will move out there. What happens to the cell in this case? Yep, shrivels up. And so this can actually cause the cell to enter into what is called plasmolysis. And this is going to be caused by water loss. Okay. So the cell, if the cell is in a hypertonic environment, I don't think that's said right. But anyways, so if this, what happens is again, is as that water can move out when the cell is in a hypertonic environment. Yeah, okay, never mind, it's fine. All right. Now, what this does is it can cause the cell to go into plasmolysis. Plasmolysis can be caused by hypertonic conditions plus desiccating conditions can cause plasmolysis. Desiccating conditions is just very dry conditions, lack of water. Think of things like the Atacama Desert in Chile. This is the driest place on Earth. And is anything growing there? No, because there's just no water. And it's so dry, it kills cells by plasmolysis. There's not even really microorganisms that grow here due to the low water content. What ends up happening as the water exits the cell, it actually can limit the protein function. Usually proteins require a hydration shell around them to function properly. If they don't have the water around them, the proteins don't fold up correctly and they don't function correctly. Even worse is as the water comes out, the cell starts to shrivel. So it starts to get smaller and smaller. And eventually it takes all this stuff that's inside the cell, gets compacted and compacted and compacted into a tighter and tighter space. And what that does is it puts physical stress and torque on things like the DNA molecules until they break apart into fragments. So my little black thing here is supposed to be the chromosome, but as the chromosome is breaking into fragments, and that is going to be lethal to cells then. Questions about that? Dry conditions can cause this. Hypertonic conditions can cause this. By the way, have you guys heard that honey is sterile? Have you ever heard that before? Okay, honey is almost completely sterile. There's a few things and some endospores that can get in it sometimes, but otherwise it's almost completely sterile. And that's because basically what honey is, it's as much sugar as you can possibly dissolve in water. Super high sugar solution, but it's a very, very, very strongly hypertonic solution. And if a bacteria falls into the honey, the water gets sucked out of the cell and it dies by plasmolysis. Questions about that?
That's why honey doesn't spoil.
Yep. And that's what we use that. We use it as a technique. High salt or high solute concentrations can act like a desiccant. They suck the water out of the cell, water moves out of the cell dies by plasmolysis. And that's why we have things like jellies, brines, and salt cured meat, for instance, as ways to preserve foods. Jellies have a high sugar content, very hypertonic solution. Brines are going to be kind of things like pickling and things like that, high salt content, but it sucks the water out of the cells. And then salt curing meat is where you just kind of put the meat with a layer of salt in it, but the salt helps protect the meat from spoiling. So, questions about that? With that being said, there are also certain species known as halophiles. Halophiles are going to be salt or solute tolerant organisms. They can survive and actually thrive in hypertonic solutions. And some of these halophiles can live up to 2 molar salt concentrations. By the way, that's a lot of salt dissolved in water, for instance. These are some salt turn ponds in California, but they're ponds with a high salt concentration. There's enough salt dissolved in the water that typically nothing lives there, with the exception of some halophiles. So there is these, you'll see that these are kind of red color, and they're red because of an Archaea species that's an extreme halophile, and it's also photosynthetic, so it has this red pigment it absorbs light energy with. And there's enough of this Archaea growing in here where most other things can't grow that it turns the ponds red, for instance. Then. Questions about that? So halophiles are specifically ones that can grow under hypertonic conditions and grow well under those hypertonic conditions, high solute conditions. All right, next parameter we're going to talk about is oxygen. Oxygen is actually pretty dang toxic. Oxygen is very reactive. And we can have some different things like different versions of oxygen, like hydrogen peroxide, which will kill things via oxygen damage. We can have bleach, which, as you guys know, you should not be exposed to, but that it bleaches NaOCl, and that oxygen that is part of it is highly reactive and damaging to nearly all molecules. And we have oxygen gas can be damaging sometimes, but the really one where we have to really watch out is what is called superoxide. Superoxide is a version of kind of oxygen gas with extra electron on there, and it is very reactive. And just about anything it reacts with gets damaged and torn apart, in essence. Oxygen in the air can be very toxic. Things like cellular respiration generates oxygen radicals like hydrogen peroxide and some others like superoxide. Immune cells actually will generate some of these and use this to kill some pathogens in our body. For instance, we have things like macrophages that will make hydrogen peroxide, and then they will secrete the hydrogen peroxide at potential pathogens to kill them. All right, we use disinfecting compounds like hydrogen peroxide or bleach to kill things, but it's killing them through oxygen damage. In particular, superoxide is the most reactive of all oxygen species. Superoxide is a version of oxygen that's very reactive, and just about anything it comes in contact with, it will damage. So if superoxide gets into our cells, and there's always a little bit of it floating around, this will react and damage all the macromolecules it comes in contact with a cell. However, the most common target for superoxide is DNA. Not because it targets DNA in particular, but because DNA is the single largest macromolecule in our cells. I've told you before how long DNA is. Do you guys remember how long DNA is? How many nucleotides joined together it is in something like an E. coli or like a human? In E. coli, it's about 3.5 million nucleotides per the DNA molecule of an E. coli. In humans, it's about 3.5 billion. So DNAs are just really big macromolecules, the biggest of all the macromolecules. Because they're so big, superoxide is just more likely to bump into it. It's just a matter of size and probability. But if superoxide bumps into the DNA, it causes double-strand breaks in the sugar phosphate backbone. It causes the DNA to literally cut into fragments, and that can be lethal. That is very difficult to repair and can be lethal to cells when it happens. Then, questions about that?
So is this like gas or like?
Yep.
Oh,
Super oxides around us all the time. Very, very, very low concentrations of it.
what if we were like exposed to a high concentration? What would,
It
like,
would be very, very bad.
would we like melt or what?
It's basically exactly the same as radiation poisoning. And in fact, technically, radiation kills you by oxygen. I'll get to that here in a second.
Are there any conditions? Like any environmental condition?
That's a good question. Oxidative stress in general, like if you are burning energy more, your cells are going to make oxygen radicals, including sometimes a little bit of superoxide.
I was like, fine.
Yes, actually, yes, high altitude will have more of it because with more UV light, sometimes oxygen will get broken down into superoxide more often.
So where do the microorganisms, sort of, like, where are they, where they like get exposed to this, to what we like?
We're all getting exposed to that. Anywhere where there's oxygen, we're getting exposed to low
Well, like,
levels.
how have we like, I guess, figured out that this damage is on?
Well, first off, you're supposed to eat a diet rich in antioxidants to be healthy. Have you ever heard that before?
Yeah.
You know what an antioxidant is?
Like, go against this?
It binds to that and basically detoxifies it. So antioxidants are anything that basically help your body get rid of oxygen radicals.
We just don't have that.
Oh, no, they have it. They have, we're gonna get to it. So different bacteria have different oxygen tolerances though. And we talked about this in lab, but this is my tubes. At the top of the tube is gonna have high oxygen concentration, down at the bottom is gonna have very low oxygen concentration. Obligate aerobes are gonna go where relative to oxygen? They require O2, only grow in the presence of oxygen. So with something like an obligate aerobe. What about strict or obligate anaerobes? So strict or obligate anaerobes, they're only going to grow in the absence of oxygen. What about facultative anaerobes? Okay, so facultative anaerobes are going to prefer oxygenated conditions, and they grow more dense up here, but they can grow without it. What about aerotolerant anaerobes? Gosh, grow independent. There we go. So they're going to grow equally well with or without O2. So they grow equally in high and low O2 conditions. Micro aerophiles grow at intermediate O2 concentrations. Okay, questions about that? All right, here's the thing. Some species, am I okay to erase all this? Okay. Obligate anaerobes, can they grow in the presence of oxygen? No, O2 kills them. Why does O2 kill them? Because they don't have these enzymes. So something like there are certain enzymes like superoxide dismutase, SOD is the name of the protein, or catalase, which is CATF. Superoxide dismutase will bind to superoxide, react with a couple protons, and it converts it into hydrogen peroxide and oxygen gas. Superoxide is really bad. Hydrogen peroxide is still bad, but not as bad. So it detoxifies that. Catalase is an enzyme that'll take the hydrogen peroxide and break it down into water and oxygen gas, even better. Okay. And for instance, obligate anaerobes lack these enzymes. Something like an obligate aerobe, I cannot spell today. It has just been a problem all day. Will have them. I've just been absolutely miserable across 2 classes in my spelling. All right, questions about that? Okay, one thing just to finish this up, working with anaerobes. If you want to grow anaerobes, you have to grow them in special culturing chambers that have no oxygen in them. You guys, so this is a anaerobic chamber. Interestingly enough, it has an airlock. So anything you want to work with, you open this up, you put it into there. You have to wait 30 minutes while the oxygen gets scrubbed out of there chemically. And then there's another airlock that you open up and you put it into there and then you start working with here. But this is a no oxygen environment for growing. Okay. If you ever forget to put, if you like have to wait 30 minutes, if you ever forget to put something into here, it is the most annoying thing ever. Because you don't realize it until you start to go do the experiment and you're like, crap, I forgot to put the inoculating loop in. So then you have to open it up, put the inoculating loop, the one thing you forgot, wait another 30 minutes before you can start the experiment and then go start the experiment. It's awful. Anyways, questions about that? All right, that's what we're in for today.